ऑन हो रहा है। और यहाँ पे भी स्टॉप एंड करना है इन स्ट्रीम। ये ठीक है।
Okay, good afternoon everyone. So today is the division class of gram-positive cocci. Okay, so gram-positive cocci, they have 4 genera, that is Staphylococcus. They have 2 families, Micrococci and Streptococci. Okay. So in Micrococci, there are Staphylococcus, and then we have Planococcus and Micrococcus. So Staphylococcus mainly causes pathogens in humans. The disease is caused mainly by the Staph aureus organism among the Streptococci. So before starting GPC, we'll make a quick revision of how to make a gram stain. So okay, so this is, first of all you have to take a glass slide. Okay, then we have to put it over the Bunsen flame to make it grease free. Okay, so after that we'll put a drop of saline and we'll pick up a single colony and emulsify it. Okay, and then we let it air dry. After air drying it, we'll put it over the flame to fix it to the glass slide. Okay, so after fixing it, we'll do the gram staining. So after doing gram stain, so gram positive organism will appear purple in color and gram negative organism will be pink in color. And so for gram positive cocci, we mainly use 2 types of media, that is blood agar and blood agar. This is the blood agar, and we use chocolate agar. So these are basically enriched medias. Okay, so to begin with the topic, gram positive cocci, they are classified into 2 families, Streptococci, Micrococci. I've already told you. So Micrococci has 4 genera: Micrococcus, Stomatococcus, Planococcus, and Staphylococcus. And in laboratories, basically we encounter with Staphylococcus, which is the most pathogenic among this. And Micrococcus is basically it's a skin commensal. So you can also encounter with the Micrococcus, and Stomatococcus, Planococcus, they are non-pathogenic to humans. So we have to differentiate mainly between the Micrococcus and Staphylococcus, because Micrococcus, as it is present on the skin commensal, they can also contaminate the samples while taking the blood sample, while taking a CSF sample, while penetrating the skin. These Micrococcus usually contaminate the samples. So to differentiate Micrococcus and Staphylococcus, we have various biochemical properties. And first of all, to differentiate, we can also use the gram stain on the basis of gram staining. So Micrococcus are relatively larger than the Staphylococcus. They are around up to 2 micrometer, and Staphylococcus they are smaller, around 1 micrometer. And the arrangement, Micrococcus has a particular arrangement, that is in tetrads. They are arranged in tetrads, and Staphylococcus, they are arranged in clusters. Okay, so other methods by which we can differentiate Micrococcus from Staphylococcus are the Hugh-Lifson method. Okay, oxidative fermentative method, where Micrococcus is because it's an obligate aerobe, so it will ferment by oxidative, and Staphylococcus are facultative anaerobes. They will utilize the carbohydrate by fermentative method. So among the Staphylococcus, Staph aureus is the most notorious and the most pathogenic, and which also produce the enzyme coagulase. So on the basis of coagulase, we can differentiate between the Staph aureus and coagulase negative Staphylococcus. So we usually encounter other cons in our lab, mainly the Staph epidermidis and Staph hemolyticus, followed by leuctinensis and all. So about the Staph aureus, so it is a catalase positive, coagulase positive, facultative anaerobe, non-motile, non-sporing, and they are occasionally capsulated. They are around 1 micrometer in diameter. So the virulence factors. The virulence factors are divided into cell-associated, toxin, and extracellular enzymes. So how do we diagnose Staphylococcus aureus? First of all, on the basis of gram stain, there will be gram-positive cocci in clusters around 1 micrometer. They will be arranged in grape-like clusters. Okay, and on nutrient agar, and then on nutrient agar, they produce golden yellow colonies. So this is one particular feature of the Staph aureus, that they produce pigmented colonies, golden yellow colonies. And on blood agar, you will see a narrow zone of beta hemolysis, because they produce the enzyme hemolysin. So because of which they have narrow zone of hemolysis on the blood agar, complete hemolysis. And on McConkey agar, will produce pink colonies because of the fermentation of lactose. And there are certain selective medias like mannitol salt agar and Rudlam's medium. So the media, this is basically the mannitol salt agar, which is a selective media for Staph aureus, where the Staph aureus will appear yellow in color, and the other Staph other than Staph aureus like cons, coagulase negative Staphylococcus, will appear pink or take the color of the media. So what are the biochemical tests? Biochemical tests to differentiate Streptococci and Micrococci are catalase. Catalase tests. First of all, the Micrococci are catalase positive and Streptococci are catalase negative. And then I've already explained Hugh-Lifson oxidative fermentative test, where to differentiate Staphylococci from Micrococci. Okay, so Micrococci show oxidative pattern, as I've already told you, because it's an obligate aerobic organism. And then we have coagulase test to differentiate Staph aureus from coagulase negative Staphylococci. So basically there are two types of coagulase test, the slide coagulase test and the tube coagulase test. Those both have different mechanism. So the slide coagulase test is mainly because of the presence of the binding coagulase, the bound coagulase, which is bound to the cell wall, and because of which these also called as the clumping factor, they cause production of fibrin between the cells. Okay, so they will appear as after like mixing it with the plasma, they will appear as coarse clumps. So tube coagulase, it has a different mechanism. Okay, so this is produced by the free coagulase. Slide is because of the bind, and tube coagulase because of the free coagulase, which is when you know this coagulase producing Staph aureus is emulsified into the plasma, they will form a clot, because it the coagulase reacts with the coagulase reacting factor present in the plasma, and then it activates fibrinogen to produce fibrins. So it will appear as like this. So this on the left side, it is the slide coagulase test, where you can see the clumps, and this is the on the right side, it is the tube coagulase. So when it's positive, it the plasma, it does not flows like this, which is given on the below. This is negative test. Okay, in positive there is clot formation, in negative there will be no clot formation. And one of the important thing about Staph aureus is the MRSA, methicillin resistant Staph aureus, which is we usually encounter in our hospitals as a hospital infection organism, a hospital infection causing organism. So MRSA basically it is produced, it is chromosomally coded by MEC-A gene, and presence of this gene, you know, the MRSA producing organism, they are resistant to all the beta-lactam antibiotics, including the cephalosporin, except for the 5th generation cephalosporin. Okay, so the drug of choice for MRSA is vancomycin. Vancomycin, though it is the drug of choice, but it is usually conserved for the most serious infection caused by MRSA. The other option we have, we can use macrolide antibiotics like clindamycin, erythromycin are better choice. So not better choice, but they are the alternatives before using vancomycin. Vancomycin is preserved mainly for the serious infections like you can say endocarditis and other serious infections. So it is a reserved drug for MRSA. So for to see the hemolysis produced on the blood agar, basically we use this 5% sheep blood agar, and it is not properly produced in the human blood agar. So talking about the catalase test, which is used to differentiate the Micrococci and Streptococci. So the principle of catalase test is that the organism that produce catalase enzyme, what they causes, they hydrolyze or they decompose the hydrogen peroxide, okay, and produce oxygen and water. So the oxygen that is produced cause this bubbling. Okay, so how do we do the catalase test? We'll take a colony and put it over the glass slide, and then a drop of hydrogen peroxide. Basically we use 3% hydrogen peroxide. We'll put a hydrogen peroxide with the cover slip, and now we'll take a colony with the cover slip, put it on the glass slide, and we'll put a drop of hydrogen peroxide and cover it with the glass slide. Then we'll see bubblings like this. That means the organism is producing catalase and it is catalase positive. Okay, so now about the Streptococci family, about the Streptococcus. So Streptococcus are basically they are divided into alpha hemolysis, beta hemolysis, and gamma hemolysis. So beta hemolysis is the complete hemolysis where there is lysis of the RBC. There will be formation of clear zone. This is the beta hemolysis. The media will look transparent. Okay, so this is because of the complete lysis of the RBC caused by the enzyme hemolysin. And alpha hemolysis mainly because of the production of hydrogen peroxide by the organism, which causes methemoglobin, which combines with the hemoglobin and produces methemoglobinemia, and this causes basically the discoloration, like brown or grayish discoloration. And gamma hemolysis is there will be no hemolysis. Now about the Streptococci, Dr. Ashwin will proceed with the Streptococci family.
Good afternoon. Coming to the family Streptococci, first of all, we are going to study about the alpha hemolytic Streptococcus, that is Streptococcus pyogenes. It is the only species under Lancefield group A Streptococcus, and it is associated with a variety of suppurative infections. For example, it can cause local infections like a sore throat, and it can even cause an infection known as scarlet fever, of which a sandpaper rash is a typical feature of this scarlet fever. It can also cause a variety of skin and soft tissue infections like cellulitis, erysipelas, impetigo, and necrotizing fasciitis. It can also be associated with a variety of diseases like toxic shock syndrome and puerperal sepsis, etc. And also it can cause bacteremia. And another important thing is that it can also trigger post-infectious non-separative complications like acute rheumatic fever and acute glomerulonephritis. Now, coming to the lab diagnosis of Streptococcus pyogenes, specimen collection and transport. We can use a pus swab, throat swab, exudates, and blood as a specimen. it has to be transported to the lab immediately. Otherwise, we can use a transport medium, which is the pipe transport media, which contains crystal violet and sodium acid. And under direct smear microscopy, we can see pus cells with gram-positive cocci, that is 0.15 to 1 micrometer in size. Now for culture, this specimen is inoculated into the various culture media for about 24 hours at 37 degrees Celsius in the presence of 5 to 10% of carbon dioxide. Now, Streptococcus pyogenes is a fastidious organism, and it does not grow under the nutrient agar and McConkie agar. Hence, it requires enriched media like blood agar, where the colonies are small, 0.5 to 1 millimeter in size, pinpoint, circular, semi-transparent, low convex with a wide zone of beta hemolysis. And there are a few selective media for this. It is crystal violet blood agar, which contains 0.1% of crystal violet, which inhibit the growth of Staphylococcus and other bacteria. And also PMF media, which is polymyxin, neomycin and fusidic acid media. Coming to the biochemical test, as first one is catalase test. It differentiates between the family of Streptococci and Micrococci. Now, Streptococci is catalase negative, and the other one is bacitracin sensitivity test. The group A Streptococcus is sensitive to bacitracin, while other beta hemolytic Streptococci are resistant. This any zone of inhibition around the bacitracin disc, that is 0.04 unit of bacitracin disc is used. Any zone of inhibition around the bacitracin disc is considered positive. Now coming to the Lancefield grouping, the biochemical identification of group A Streptococcus can be further confirmed by this Lancefield grouping. It is very useful in these epidemiological study settings, and it is based on the C carbohydrate antigen which is present in the cell wall. This C carbohydrate antigen can be extracted from the cell wall by using a variety of methods, like using hydrochloric acid, by using formamide, by using autoclave, or by using special enzymes which are obtained from Streptomyces albus. Now the extracted antigen is tested with a commercially available group-specific antisera by lattice agglutination test. That is the Streptex test. This is about Lancefield grouping, and the Lancefield identified 20 serogroups of beta hemolytic Streptococci, which was based on the C carbohydrate antigens in the cell wall, and it was grouped into A to V except I and J. Now Streptex is a commercial kit available for the grouping of beta hemolytic Streptococcus. Streptex is a rapid lattice agglutination test, and it uses simple enzyme extraction method is employed in this test. Now, Streptococcus produces an enzyme, a toxin known as streptolysin O. It is an exotoxin. Now, we can employ lattice agglutination test to detect the antibodies in the serum which is produced against this streptolysin O. Hence, it can be used in the diagnosis of non-suppurative sequelae of Streptococcus pyogenes infections like rheumatic fever and acute glomerulonephritis. A positive test is indicated when the value is more than 200 international unit per ml. Now, coming to the Enterococcus. Enterococcus was initially grouped under group D Streptococcus, but later classified as a separate genus because both of them answer bile esculin hydrolysis test as positive. Now, this Enterococcus can cause a variety of infections like urinary tract infections. It can cause late onset neonatal sepsis and meningitis. It can cause intra-abdominal pelvic soft tissue infections. It can lead to bacteremia and mitral valve endocarditis. Now, on lab diagnosis, these are gram-positive oval cocci which are arranged in pairs, arranged at an angle to each other. That is a particular shape of arrangement, that is spectacle shaped arrangement. And in the blood agar, we can see non-hemolytic transclusion colonies, that is they are showing gamma hemolysis, that is there is no hemolysis. And in the McConkie agar, we can see minute magenta pink colored colonies, and bile esculin hydrolysis test is positive. Bile esculin hydrolysis test means that they grow in the presence of 40% bile and hydrolyzes esculin to esculatin, which combines with ferric chloride to form a black colored complex. And also the PYR test is answered by Enterococcus. They can grow in the presence of extremes of conditions like 6.5% sodium chloride, 40% bile, and pH of 9.6 and varying temperatures between 45 degree and 10 degrees Celsius. Now, for a bile esculin hydrolysis test, there are two methods to do this test. That is, one is tube method and the other one is plate method. In tube method, we use 0.5 ml of sodium, 2% sodium deoxycholate in 1 ml of emulsified colonies in normal saline. And the turbidity detected is between 0.5 to 1 McFarlane units. And in the plate method, we use 10% sodium deoxycholate directly on the colonies, and after half an hour we can see there is a clearance of the colonies due to this is the bile esculin hydrolysis test, and this is the appearance of Enterococcus on the blood agar. Now coming to the pneumococcus, Streptococcus pneumoniae, and it is the leading cause of lobar pneumonia, otitis media and meningitis of all ages. Actually, this is a capsulated organism, hence the risk factors like asplenia, splenectomy, sickle cell anemia and other hemoglobinopathies, the age less than 2 years and other comorbid conditions increase the risk of pneumococcal infections. Now coming to the lab diagnosis, these are fastidious organisms, hence do not grow in nutrient agar or nutrient broth. Simple gram staining will demonstrate the lanceolate or flame-shaped gram-positive cocci about 1 micrometer in pairs, which is surrounded by a clear halo. It is due to the presence of a capsule. Now, in CSF specimen, capsule can be better demonstrated by using Indian ink staining technique. Now, in the blood agar, after 18 hours of incubation,
gram stain is very important for CSF. In case of meningitis, so that they can provide the empirical antibiotic.
In the blood agar, after 18 hours of incubation, the colonies are small, that is 0.5 to 1 millimeter dome-shaped, glistening, surrounded by greenish discoloration due to alpha hemolysis. On further incubation, the colonies become flat with a raised edge and a central depression. It is due to the autolysis of the central colonies by the autolysin enzyme. Hence, we get a double ring arrangement, that is Drotman-shaped arrangement or carom coin-shaped appearance when we are looking from the above. And in the chocolate agar, they produces a greenish discoloration due to a bleaching effect. And also coming to the biochemical identification of Streptococcus pneumoniae, it is important to differentiate it from other alpha hemolytic Streptococcus, that is Streptococcus viridans. First of all, solubility in the bile. Streptococcus pneumoniae is soluble in the bile because due to the enhanced solubility of Streptococcus pneumoniae in the presence of bile. And optocin sensitivity is answered by Streptococcus pneumoniae. They are sensitive to 5 microgram of optocin disc, but Streptococcus viridans is not sensitive. Now the inulin fermentation, Streptococcus pneumoniae ferment inulin to produce acid, while Streptococcus viridans does not. And another method is to demonstrate the capsule is Quelling reaction, where we use we can demonstrate the capsular swelling by using a type specific antisera which is raised in the rabbits and using simple dyes such as methylene blue directly on the colonies under on a slide under a microscope. Now for the Streptococcus pneumoniae, there are two vaccines available. One is pneumococcal polysaccharide vaccine, which is 23 valent, and the other one is pneumococcal conjugate vaccine, which is 13 PCV 13. Thank you.
Stop streaming.
I don't know.